on hormones. And I hope that within 2 lectures, we should be done with the hormones too. So today and then the next lecture, we should be done with hormones and move on to the next. So gradually, we are dealing with our plan for the semester, and I hope that we'll finish maybe within the next 4 lectures. We should finish all the things that are on our plan before your exams. Now, imagine that we don't have any form of communication. I mean, let's see how important communication is. That we are together in a society. I don't have a way to communicate with you. Like there is nothing like I can talk with you. I can't, as I'm here, I'm talking, you can hear me, you can listen and you understand what I'm saying. Imagine that all this system is not there, that you can communicate. Even if you are together, there's nothing like language, not even sign language or anything. You see, it will be total chaos. There won't be anything like that. So we see communication to be that important. It's the same way to do with the biological system, that you have communication systems within the biological systems. I mean, generally, in biological systems, there are two major forms of communication, either by chemical communication, that is to have some chemical substances released which will generate some response, or we have electrical signals that go along nerve cells. And of course, those electrical signals will also result into secretion of some chemical compounds, say neurotransmitters, which will continue to give the particular communication across the synapses of the nerve cells. So we have these communication systems by electrical signal or by these biological compounds, biomolecules in the body, usually neurotransmitters. But we also have the hormones that also, regulate biochemical reactions. And generally, we have, say, a signaling cell that is releasing some biomolecules, what we refer to as ligands, to a target cell. And you can have on the surface of the target cell a receptor response in that target cell. So even though we're talking about biological system in the sense that, say, if you take the human body, that you are having a lot of these, cells, signaling cells that can produce some ligands released, and those ligands will cause some biochemical response in a particular cell as some form of a biological response. It is actually also for survival, because another thing is that this is a communication system by which cells recognize some intrusion of, say, a pathogen, a virus, or some bacteria, and they begin to respond to this intrusion, so that they can give signals to, say, the immune system. So you can have some immune cells fighting this bacterial invasion or viral invasion. Even when you take unicellular organisms, I mean single cell organisms, that if you remember, you take, let's say, protozoa, or I think you did things about, how do you call it, paramecium, and you take even bacteria that is in a colony. I mean, you have such kind of communication with the organism with its environment. You have such cellular communication between the organism and its environment. So yes, cellular communication is that important in these aspects. Now we'll continue to take a look at the different kinds of cellular communication that we have. The first we are looking at is autocrine signaling. Autocrine signaling. So what we are doing is, even though, if you see the topic, it says hormones. So the hormones are signaling molecules. But before we come to talk about the hormones themselves, we need to establish some foundation on, say, cellular communication, the mechanisms by which these signaling systems, the communication systems work, before we dive into the hormones specifically, looking at the hormones that we have, looking at a particular system, the endocrine system, where that is responsible for secreting these hormones. So now we're looking at the autocrine signaling as one of the forms of cellular communication. By this, you have a cell that is signaling itself. So what you see, the diagram you are seeing on the left is that there's signaling molecules released from a cell, and those signaling molecules go to bind onto a receptor on the same cell, and the response is to trigger some transcription and translation of some genes in the nucleus. You find this type of signaling in the immune system. So we'll take a look at some specific example of this, and you have this in, say, what we refer to as the macrophage immune response. So that when a viral or a bacterial invasion is recognized, you have these immune cells releasing some signaling molecules, cytokines, like interleukins, that are going to bind onto a receptor on the same cell, and the immune response will be that there will be transcription and translation of some specific genes, and that will be some proteins, and those proteins will be some enzymes that will catalyze some specific reactions in response to that pathogen invasion. You will see this also in, say, early embryo development. I mean, when you have cells dividing at a very fast pace, you find some autocrine signaling in those cells as well. Similarly, you see such a thing occurring in, say, cancer cells, where there is this cell proliferation is a way of these cancer cells signaling themselves to reproduce them more, and that results into formation of tumors. So yes, you have this autocrine signaling, which is for signaling self. The cell, a particular cell signals itself that way. Another form of communication that you find, this is particularly say in the brain, so in the brain, and also in cardiac cells, in the heart, you have this form of communication, what you refer to as the gap junctions. It's a very fast way of movement of ions between these, like in the form of electrical signals between the cells. So the cells have these channels, these ion channels, which is made the membrane. Within the membrane, you have this hexameric protein, what we refer to as the connexon. So if you look at it, it's divided into six. So it's 6 proteins coming together to form a channel. So it's a hexamer. So you have that channel, and those channels between two cells actually join, and you have ions passing through these channels between the cells. Each of these proteins we refer to it as connexin. So in place of the O is I, connexin, but together is connexon. And so you have this joining, and that will form a gap junction. Now, another type is the paracrine. The paracrine signaling. So the molecule, and that signaling molecule will be acting on a nearby cell. So it's the signal is not going that far. It's not going to move into a different tissue somewhere, but it's a nearby cell that the signaling molecule is targeting. So, for instance, say if we take the pancreas, pancreas secreting, say, insulin, it will be acting on some nearby cells, to trigger some reactions in order for blood glucose to be controlled. So you have such paracrine signaling also as one of the forms of cellular communication. So I mean, you have, when I say the pancreas, of course, specifically what you refer to as the islets. So these are some particular, tissue cells that are responsible for secreting these hormones directly to nearby cells. Then the endocrine. This is actually what we are going to focus on, the endocrine system. In the end, this is what you will focus on. Now, you see that for endocrine signaling, you have an endocrine cell. So let's say that's a gland that secretes a signaling molecule, which is a hormone, into the bloodstream. And that signaling molecule flows to a different to those hormones. So that is endocrine. So you see in endocrine, the signaling molecule is secreted into the bloodstream, and the signaling molecule flows to a different tissue. So the biochemical response is going to be in a different tissue that is different from where the signaling molecule is being secreted from. So that is endocrine signaling. And we are going to focus on the endocrine system. So this is the So I said from the beginning, we are just laying a foundation to understand certain fundamental things before we go into these hormones, themselves, and looking at mechanisms of action and all of that. Now, a receptor that is on the surface of the cell. Sometimes, so usually you can have it as not just a protein, but a glycoprotein. So that's a protein which is in complex with the carbohydrates. You can even have lipoproteins. So a protein which is bound to a lipid because layer. And within that phosphobilayer, you can have some transmembrane proteins. And you have some of these transmembrane proteins which are responsible for cell recognition. And these, they have this recognition for specific ligands. So if a specific hormone will have some specific receptors that are going to respond or they are going to bind to that ligand. Also referred to a hormone, which is a signaling molecule, as a ligand. And a receptor is a protein which is either embedded in the cellular membrane that binds with a ligand. And when it binds with a ligand, it then triggers a biochemical response within the cell. And you can have either these transmembrane protein that I referred to, or you can have what we refer to as internal receptors. Those ones are inside the cell. What it means is that for those ones, the ligand will have to go into the cell, will penetrate through the cell membrane into the cell to bind to those receptors, which are the internal receptors inside the cell. So you have those two types of receptors, the cell surface receptors, which are also known as transmembrane receptors, and you have the internal receptors. Then you also know that these receptors are specific to a ligand. They will be binding to specific uh, signaling molecule. Okay, so those things are clear now. So that you will understand when we continue to look at mechanisms of action of these hormones. You will know why a hormone is interacting with a cell surface membrane, or it is penetrating into the cell to now bind to a receptor in the cell to trigger a biochemical response. We have hydrophobic hormones, which we also refer to as group 1 hormones. And the hydrophobic hormones are steroid hormones, thyroid hormones, and we also have the calcitriol. Now, when you take a look at this, the steroid hormones is having that sterol nucleus. So it's usually these are the sex hormones, estrogen, androgens. You have them as steroid hormones. Thyroid hormones, if you pay attention to this structure, you realize that this is a derivative of the amino acid tyrosine. It's a derivative, but it's hydrophobic. Then you have calcitriol. Calcitriol, if you pay attention to the structure, you realize that it's very much like a steroid hormone, except that the second ring of the steroid, if you look at it, the second ring of the steroid is opened. So these three types of of hormones, which are classified as hydrophobic or group 1. You remember we just said that the cell membrane is a phospholipid bilayer. So it's a lipid. All right. So if it's a lipid, it means that a substance that is lipophilic. In other words, this word, hydrophobic. Hydrophobic will mean that that substance will be lipophilic. It means that these hormones can easily pass through the cell membrane into the cell. So these ones will be acting on internal receptors. Okay, now we can take a look at some of these group 1 hormones. And we see the sex hormones, estrogens and androgens. I mentioned earlier, these are some types of hydrophobic or group 1 hormones. Then you see, what you see here is just, say the hormones are in the first column. And then in the second column, we have the tissues that secrete these hormones. And then in the third column, you have the functions that these hormones carry out. So for instance, if you look at estrogens, these are secreted by the ovaries and the adrenal cortex of the kidneys, and then you have them responsible for female sexual characteristics and also regulating the menstrual cycle. You have the progestins also being secreted by ovaries and the placenta. These are also involved in regulation of the menstrual cycle and the maintenance of pregnancy. And in males, you have androgens, which are secreted by the testes and the adrenal cortex. These are responsible for male sexual characteristics and the process of spermatogenesis. Then you have the glucocorticoids and the mineralocorticoids. These are also secreted by the adrenal cortex of the kidneys. The glucocorticoids, they regulate different metabolic processes. That is metabolism of carbohydrates, lipids, proteins. You see, when we are looking further, you see them acting in different metabolic processes, and also in the immune. And also water balance in the body. You have the calcitriol, also the kidneys, and that are secreting them. They promote absorption of calcium from the intestines, and also from the kidney and from the skeleton, the bones, the bone marrow. It can be triiodothyronine or tetraiodothyronine, and these are secreted by the thyroid glands. As we go ahead, we will see more of this, where these glands are located, and we'll talk more about them. And the thyroid glands also, they are also responsible for regulation of different metabolic processes, regulating metabolism of, say, carbohydrates, proteins, lipids. You see them acting in those reactions. All right, so that is hydrophobic. So if you haven't kept anything at all, you should understand that the hydrophobic hormones are able to penetrate through the cell membrane into the cell to bind to internal receptors because they are hydrophobic, which means they are lipophilic. And the cell membrane is a phospholipid bilayer. All right. So now, with that, you can see clearly from the mechanism of action. I think we've seen this before when we were perhaps looking at a process of transcription, translation, maybe. So we see a hormone, which is a triangle with an H in it. And that hormone, remember, it said what? Is hydrophobic, which it goes into cytoplasm, and in the cytoplasm, it binds to the receptor. So the receptor is depicted here with a letter R. And so you now have the hormone receptor complex. Now that hormone receptor complex also can penetrate through the nuclear membrane into the nucleus. And in the nucleus, they bind to a promoter region of a gene. And that activates the process of transcription of that gene. The gene is transcribed to a messenger RNA. The messenger RNA comes out into the cytoplasm and undergoes a process of translation. A specific protein is synthesized, and that specific protein will then carry out the biochemical response, because that specific protein can be an enzyme that will trigger a reaction, perhaps even a cascade of reactions, one reaction after the other, until the final biochemical response is achieved. Okay, so that is let's take the hydrophilic hormones. The hydrophilic hormones. Now, from the word clearly, these cannot penetrate through the cell membrane, which is a phospholipid bilayer, because this is hydrophilic, say water soluble. These can either be peptide hormones or amino acid derivatives. Now pay attention and realize that there's a difference between it must contain at least 2 amino acids to be a peptide. But if you say an amino acid derivative, it means it's a single amino acid that has a modification to its structure. So you have peptide hormones like insulin, glucagon, oxytocin, antidiuretic hormone. All of these are proteins. So they are peptides. These are peptide hormones. But you have some amino acid derivatives. Well, the structures you see here are some amino acid derivatives, what we also refer to as catechol amines. Catechol amines, because you see the structure, the 6-sided ring with the 2 hydroxyl groups on it, that structure is catechol. And so if you see a hormone like, say, dopamine, you can see it's just an attachment of some ethylamine on that gives you the structure of dopamine. So it's a catechol with an amine group in it. So that's a catechol amine. Same thing if you look at noradrenaline or norepinephrine. You see, same thing. The only difference you see between the structure of dopamine and norepinephrine is that on this carbon, there is a hydroxyl group, which is in blue color. That you don't find in dopamine. Similarly, if you take a look at epinephrine, which is also known as adrenaline, you see the difference between noradrenaline and adrenaline is that there is a methyl group on the amine group there, which is not in noradrenaline. So these are amino acid derivatives. Which amino acid will be these? Obviously, this will be tyrosine. So if you look at the structure of tyrosine and take a look at this, clearly is a modification of the structure of tyrosine that gives you these hormones, these hydrophilic hormones, the catecholamines. Right. So here too, the hydrophilic hormones, they are divided into three main groups. So if you have hydrophilic hormone being group 2 hormones, remember that these group 2 hormones can be divided also into 3 subgroups. And the first subgroup is what we are looking at. This group of hormones make use of cyclic adenosine monophosphate as a second messenger. Now remember that the hydrophilic hormones cannot penetrate through the cell membrane into the cytoplasm. So they will bind onto receptors on the cell surface. And when they bind to receptors on the cell surface, they trigger reaction inside the cell. So the message that the hydrophilic hormone is carrying is transferred to a molecule inside of the cell to carry out the biochemical response. And that transfer is through the cyclic adenosine monophosphate. That's why it's being referred to as a second messenger. So we refer to these group 2 hormones, this subgroup of group 2 hormones, as cyclic adenosine monophosphate. Adrenocorticotropic hormone, a number of them, follicle stimulating hormone, luteinizing hormone, chorionic gonadotropin, thyroid stimulating hormone, beta endorphins and enkephalins. All of these are secreted by the anterior pituitary gland in the brain. You have them there as some stimulating hormones. And you see, the first you can see it stimulates the release of adrenocorticosteroids. So in the brain, you have that hormone secreted and it will stimulate the release of a particular hormone. And you see similarly, the follicle stimulating hormone, this you find in both males and females, and you have in females, it will stimulate ovulation. So that is in the menstrual cycle, ovulation and estrogen synthesis. So it will control also the synthesis of estrogen. That's another hormone. Then you have in males, it will be facilitating the synthesis of the spermatozoa. That's the process of spermatogenesis. Luteinizing hormone will be stimulating synthesis of estrogens and progesterone. Here too, will be regulating the process of ovulation. So that will be in females. And it also promotes androgen synthesis in the testes. So that will be in the males. Then we have the chorionic gonadotropin hormone. This stimulates progesterone release from the placenta. So that will be in pregnancy. And you have the thyroid stimulating hormone. Of thyroid hormones from the thyroid glands. Beta endorphins and enkephalins. They act like, what we refer to as natural analgesics or pain relievers. We have this antidiuretic hormone, which is responsible for regulating water balance. You have that from the posterior pituitary, and is responsible for reabsorption of water by the kidneys. Glucagon by the pancreas. This one, I think it's a common one, It does the opposite of what insulin does. So it goes to increase blood glucose level, stimulating glycogenolysis. So carbohydrate, which is stored as glycogen in the liver or in the skeletal muscle. Usually it is in the liver, because skeletal muscle, it is when you are involved in a vigorous activity that you have that process of glycogenolysis. And also the breakdown of lipids, lipolysis. So when energy is needed, yes, glucagon triggers that process of the breakdown of glycogen. And you have the parathyroid hormone, which will be from the parathyroid gland. This will be increasing calcium levels in blood because it promotes the release of calcium ions from the bones. You have calcitonin from the thyroid glands, which is doing the opposite. So you see, regulation. It's doing the opposite of what the parathyroid hormone is doing. It lowers the serum calcium. It decreases it. So it decreases the uptake of calcium by the bone and the kidneys. Then we have epinephrine and norepinephrine. That's the adrenaline, noradrenaline, coming from the adrenal medulla, also of the kidneys. And, this adrenaline is, and noradrenaline, they are referred to as the fight or flight hormone. So that at a point where the body is needed to, react to some threat or run away from it, you see a lot of these hormones secreted. And adrenaline, which is the epinephrine, increases heart rate and blood pressure, promotes glycogenolysis, again, breakdown of carbohydrate storage in the liver and in the muscles, as well as breakdown of lipids in the adipose tissue, which is lipolysis. And norepinephrine also does that, stimulates lipolysis in the adipose tissue. All right, so these are hormones that are dependent on cyclic adenosine monophosphate. We'll take a look at the mechanism of action of these hormones. So here too, I think maybe we've seen this before, except that maybe we didn't link it directly to the hormones, or maybe it's not in your class. And we see this from the top, you see a hormone. The hormone, binds to a receptor that's in the cell membrane, because the hormone cannot penetrate into the cell. Remember, the hormone is hydrophilic. So it binds to the receptor, and that receptor is bound to some protein, which is in complex with GTP, guanosine triphosphate. That is a cellular form of energy. And upon its binding, actually, you have this protein getting dissociated, you have, say, some dephosphorylation occurring. It's a form of a trigger, carry out the message. And what happens is you have this enzyme, adenylate cyclase, which now catalyzes the conversion of ATP to cyclic adenosine monophosphate, just by the removal of two phosphate group, and the monophosphate undergoes cyclization to have cyclic adenosine monophosphate. And you see that cyclic adenosine monophosphate can bind to regulatory portions of an enzyme to dissociate those regulatory portions. And the catalytic portions of the enzyme then go to trigger another reaction, which is phosphorylation of a protein. So that's call it a protein kinase. What you see as CC is a protein kinase. It goes to catalyze the transfer of a phosphate group from ATP onto a protein. Obviously, that protein is going to be an enzyme, activating it into a phosphoprotein. And that enzyme then carries out the ultimate biochemical response. Of course, after it carries out the ultimate biochemical response, it goes back to its original form, where another enzyme, phosphatase, does the dephosphorylation of that enzyme. So you see the cyclic adenosine monophosphate dependent hormones acting in this way, where the second messenger is the cyclic adenosine monophosphate. Right. We can take a look at the action of some of these hormones. So we have here epinephrine, norepinephrine, glucagon, thyroxine, glucocorticoids, thyroid stimulating hormone, adrenocorticoid hormone, growth hormone. All these hormones activate the process of the conversion of ATP to cyclic adenosine monophosphate, all these hormones. And you see a hormone like insulin. In addition to some vitamin B's, this vitamin B, niacin, vitamin B3, and then a progesterone. You have this insulin doing the opposite of, of course, you can see glucagon in this group. So insulin is doing the opposite. Insulin is not going to, is going to block that conversion of ATP to cyclic adenosine monophosphate. And what do you see after the formation of cyclic adenosine monophosphate? Insulin again so that once it's not cyclic adenosine monophosphate, it will not be active to carry out the reaction. Here too, you see caffeine. Caffeine also inhibits that conversion of cyclic adenosine monophosphate to its non-saccharide form, which means caffeine promotes the formation of cyclic adenosine monophosphate. So you come to see, this is a biochemical explanation for why some of these caffeine-containing products, like some teas, some once the caffeine blocks this reaction, it means that cyclic adenosine monophosphates will be readily available, to activate a protein kinase, which is responsible for the conversion of an inactive triacylglycerol the ultimate biochemical response. The ultimate biochemical response is for the breakdown of triacylglycerol into 3 fatty acids and glycerol. I mean, you've been saying that fats and oils are broken down into fatty acids and glycerol. I mean, we've been talking about this in terms of digestion. So here you are looking at it. It's occurring in 3 steps. You have the first free fatty acid being removed from triacylglycerol. You are left with diacylglycerol. Then the second free fatty acid is removed. Then you are left with the monoacylglycerol. And the last free fatty acid is removed. So you have the 3 fatty acids and then glycerol at the end. So you can see how the hormone formation of cyclic adenosine monophosphate, which further activates a protein kinase, which carries out that transfer of a phosphate group from ATP onto a particular enzyme, triacylglycerol lipase, to start that reaction. Because lipase, then of course, the second enzyme is going to be diacylglycerol lipase. And then the third enzyme is going to be monoacylglycerol lipase. So you only need the hormone to trigger, to activate Then we can also now look at a second group. We said there are the group 2 hormones are divided into 3 subgroups. We were looking at the first subgroup, which was the cyclic adenosine monophosphate dependent. Now we have moved to the second subgroup, which is making use of phosphatidylinositol in combination with calcium as second messengers. So here, and you see here the hormones, thyrotropin releasing hormone released by the hypothalamus in the brain, and it promotes the thyroid stimulating hormone release. You have the gonadotropin releasing hormone also in the hypothalamus, stimulating the release of follicle stimulating hormone and luteinizing hormone. You have gastrin, which is secreted by the stomach, and this is more or less a digestive hormone, and this stimulates gastric hydrochloric acid and pepsinogen secretion for the metabolism of proteins. And you have cholecystokinin also secreted in the intestines, and this stimulates contraction of the secretion of bile into the duodenum in order to promote the metabolism of lipids and secretion of pancreatic enzymes. And these hormones are classified as this phosphatidylinositol calcium dependent. So let's see how this group of hormones, how they act through phosphatidylinositol and calcium. So from the top, we see phosphatidylinositol undergoing phosphorylation. So of course, that phosphorylation is occurring in the cell membrane. Remember, the hormone cannot penetrate through the cell membrane. It is hydrophilic. So the first reactions that will trigger further reactions in the cell are going to be occurring in the cell membrane. And you see the phosphorylation of phosphatidylinositol converts it into phosphatidylinositol 4,5 biphosphate. So 2 phosphate groups are attached on that phosphatidylinositol, which means from there, if you have a cleavage of the inositol biphosphate from that phosphatidyl, let me say, you have a diacylglycerol. That is a glycerol with 2 fatty acids on it. And then on the last carbon, you are having what you refer to as the inositol and the 2 phosphate groups. So when that is cleaved off, you'll be left with a diacylglycerol. Now, what it means is that what is cleaved off also undergoes a further phosphorylation, and you have inositol 1,4,5 triphosphate. Remember from here it was a biphosphate, phosphatidylinositol 4,5 biphosphate. But when it's cleaved off with further phosphorylation, you have inositol 1,4,5 triphosphate. Now you see the diacylglycerol can activate a protein kinase. And what do protein kinases do? Kinases are responsible for the transfer of phosphate group, particularly from ATP onto other biomolecules. And that is actually going to be a phosphorylation of another protein, which is an enzyme. So it's a cascade of reactions, one reaction after the other, until the ultimate biochemical response is achieved. So you have this activation of a protein kinase, which when it goes into phosphorylation of an enzyme, that enzyme then goes into a particular reaction, catalyzing it. Or you have this inositol triphosphate releasing, activating the release of intracellular calcium. So we just saw calcium can be released from places like the bone marrow, the kidneys. So once that is released, that calcium actually acts as a cofactor to the protein kinase. So that's why we are saying that, in the name we say what? Phosphatidylinositol calcium dependent, calcium ion dependent. And that calcium can also just directly go to the regulation of other enzymes. We can see it more colorfully. Let's see it more colorfully here, so that perhaps this one will make it much more clearer. We see there you have this phosphatidylinositol, biphosphate. So you see what I was saying. If you look at it, you see that you have 2 fatty acids that are bonded on the glycerol backbone. And then you have this phosphate group with inositol. So a hormone acts on this enzyme, phospholipase C. And when that happens, the phospholipase C cleaves off the inositol biphosphate, and that inositol biphosphate gets phosphorylated along the line and also becomes inositol triphosphate. That's what we saw earlier. So that's what we are seeing, the inositol triphosphate. And what does the inositol triphosphate do? This inositol triphosphate binds to calcium ion channels that are embedded in the endoplasmic reticulum membrane. And these calcium ion channels open up. When they open, calcium ions are released from the endoplasmic reticulum into the cytoplasm. And these calcium ions, they are cofactors. They serve as cofactors. They bind to a protein kinase, and that protein kinase goes back to bind to the diacylglycerol, which is formed when you have the inositol biphosphate cleaved off from the inositol, the phosphatidylinositol biphosphate. And so then you have the protein kinase. Now with a cofactor, the calcium ions can then now catalyze phosphorylation of a protein, what we are referring to as a substrate. That's an activation of that enzyme, which will carry out a particular biochemical response. All right. Now, we take the third group. That's a third subgroup. The third subgroup of the group 2 hormones. These ones, they are not making use of cyclic adenosine monophosphate or the phosphatidylinositol in combination with calcium as the second messengers, but they use several intermediates as a second messenger. I think even some of them will make use of the same mechanism which we have looked at. I think insulin will do that with inositol triphosphates, the diacylglycerol calcium system, which we just looked at. This last pathway, we won't look at it. It's just so complex. This is also some protein kinases, but you have lots of things happening over there. But we can take a look at this, the JAK start, and take a look at it. And we see growth hormone is one of those. Prolactin, these two, they are released from the anterior pituitary. You have the growth hormone promoting the growth of bones, organs. I mean, for elongation of the bones, particularly in children growing up to a certain point where growth stops. But of course, the growth hormone will always be acting. You have even in times of female secreted by the posterior pituitary, stimulating uterine contraction. So that is in labor. In childbirth, you have that. Oxytocin is responsible for stimulating uterine contraction, as well as milk ejection. So that is after labor, in milk production or milk ejection that is contraction of the mammary glands for the newborn baby. You have insulin, of course, you know this is secreted by the pancreas. And you also know the function, lowers blood sugar. So what we refer to as a hypoglycemic effect, and it promotes protein synthesis, lipogenesis. You have somatomedins, which are insulin-like growth factors, or some other growth factors, IGF-1, IGF-2. These are produced in the liver. They are all growth related. They have growth related functions, and yes, they stimulate also not only the growth of the bones and the organs, but also cartilage that is providing cushioning at the joints. So we can take a look at this JAK-STAT mechanism. Now, I mean, from the name JAK-STAT, the STAT is signal transducer and activator of transcription protein. That's STAT. So it's for transducing. That is signal transduction only means signal transportation, and of course transcription. So to activate transcription of a protein, transcription of a gene, which will result into messenger RNA, and then from the messenger RNA you can have translation. That's what it means, activator of transcription. And we see this a receptor, which is so at the top here, where the color is teal. Well, you can say it's blue. Some people will see it as green, and below it is sort of pale yellow. And you have this cell membrane. So inside the cell membrane, you have this protein, the cytokine receptor. And that cytokine receptor, when it attaches to cytokine, which is the molecule which brings, that messenger molecule, the cytokine, or say the ligand, which comes to bind onto the receptor. So now we see it binding onto the receptor in the cell membrane. And what you see is that the Janus kinase, catalyzes phosphorylation of the cytokine receptor. So you see those phosphate groups are attached. And a second phosphate group also comes together with the STAT. That is the signal transducer and activator of transcription protein. Then you see the next thing is that, so these are monomers. These are protein monomers that eventually form a dimer. So you have a dimer of the sort which dissociates. So this dissociates from the receptor and go into the nucleus. This can penetrate through the nuclear membrane, and there will activate the process of transcription. So you see why we say it's a second messenger. The cytokine, which is the first messenger, is not able to penetrate through the cell membrane, but you see that you have these molecules, the Janus kinase and the STAT, working together to carry the message to the nucleus. There, it will trigger transcription of a particular gene. We can take a look at it much more closely with a specific example with coronaviruses. So for instance, you have this coronavirus that has attacked a cell, and you see that you have a receptor for cell recognition. So it recognizes that coronavirus. When it recognizes the coronavirus, it triggers some interleukins, cytokines, binding onto the receptor embedded in the cell membrane. There is process of activation. And so you see the JAK-STAT complex, which is on the receptor. After you have the cytokine bound on the receptor, and there is an activation, what you see is that there is that dissociation of this dimer. The STAT, phosphorylated dimer, moves into the nucleus through the nuclear membrane, binds to a promoter region of a gene. Then that gene is activated, and you have transcription of the structural genes. That will be some particular protein, and that protein will move out of the cell, into cytokine storm. You have macrophages, T cells responding in that case of invasion, viral invasion of the cell. So they begin to fight. That's an immune response. So here too, yes, that's a hormone that cannot penetrate through the membrane, the cytokine. It is hydrophilic. All right. We can have, I think we've packed our brains with a lot of mechanisms today, but we can lay some foundation for the next lecture, where we are going to take the endocrine system. What we're looking at is the glands, the tissues that are responsible for the secretion of some these hormones that we've talked about. We see the pineal gland is in the brain, the pituitary gland also there in the brain. So we are looking at it from top coming down. Then we have we have the adrenal glands, which are at the anterior position of the kidneys, the pancreas, ovaries in the females, and then testes in males. These are responsible for the secretion of sex hormones. So these are we are going to be looking at specific hormones that are secreted from each of these glands, and look much closer to their functions. Perhaps look at maybe their deficiency, what it results to, and things like that. We'll zoom in from our next lecture. We'll zoom in on each of these hormones from each of these glands. We can take a look at the structure of the endocrine system in general. And that is, you have the hypothalamus in the brain. I mean, that is, well, even though itself is not a gland, it's part of the brain, but it's referred to as a master gland of the endocrine system. So here, if you take uh, you pay attention, pay a close look attention to this, you realize that all the hormones that are secreted by the hypothalamus are releasing hormones. What it means is that they only trigger the release of another hormone. So that's what they are doing. So you see that you have the anterior pituitary, which is under the regulation of the hypothalamus. Only the anterior pituitary. You see the posterior pituitary is not under the regulation of the hypothalamus. It's on its own. And what you see is the hypothalamus. It has this, you have the thyroid releasing hormone. Then you have the corticotropin releasing hormone. See, all of them are releasing. And you have this gonadotropin releasing hormone. You have growth hormone releasing hormone. And you have also growth hormone releasing inhibiting hormone, which is doing the opposite of the growth hormone releasing hormone. Then you have the prolactin releasing inhibiting hormone. So all of these are just acting on the anterior pituitary, I mean, giving a signal to the anterior pituitary to secrete, or to act on some specific glands. The thyroid hormone releasing hormone, you see that when it acts on the anterior pituitary, the anterior pituitary secretes thyroid stimulating hormone. So itself too is not yet, the anterior pituitary has not, released the thyroid hormone, but it's a stimulating hormone that acts on the thyroid gland. And then the thyroid gland secretes thyroid hormones, either triiodothyroid hormones or tetraiodothyroid hormone. You have the corticotropin releasing hormone. When it acts on the anterior pituitary, it secretes adenocorticoid stimulating hormone. And you have that acting on the adrenal cortex. And then the adrenal cortex, that is in the kidneys, then these will now secrete adrenocorticosteroids. These are the hormones. Similarly, you have the gonadotropin releasing hormone acting on the anterior pituitary, which will act on the anterior pituitary, which will release luteinizing hormone. That luteinizing hormone will act on the ovaries and the testes. Same way, you have follicle stimulating hormone also acting on the ovaries and testes. And what you see is that you have these now, if it's androgens in males. So in males, you have the testes releasing androgens. In females, the ovaries releasing inhibiting hormones. So these two, they regulate growth They will act on the anterior pituitary to release growth hormone. And that growth hormone can act directly on, say, bone for bone elongation, that is in growth, or you see it acting on the liver, where you have, that is specifically for this growth hormone the opposite of the growth hormone, where so there's that regulation. So there isn't an overgrowth. And you have the prolactin releasing inhibiting hormone also controlling the release of prolactin, which acts on the mammary glands for the contraction of the mammary glands for ejection of milk. And you see these estrogens, androgens, of course, acting about a regulation of the hypothalamus. What you see is it secretes antidiuretic hormone, which regulates water reabsorption in the kidneys, and oxytocin, which is acting in child labor, for uterine contraction. So this is the general structure of the endocrine system that we will look at. The next time when we meet, what we do is we'll take each and every one of these hormones and zoom in and see their function and possible maybe disease conditions that are associated with them, and maybe even open if you have any questions, any contributions, anything you want to share with us. You can either write in the chat or you raise your hand. I will enable your microphone and all right, so I see Joshua Balija. Let me unmute you.